University for many years. In fact, we've even worked in the same department at times, haven't we?
We're colleagues.
But Robert's going to be talking to us about one of his special areas of expertise, and I won't delay further. I'll hand over to you, Robert.
Thank you very much, Gordon, and thank you very much to Society, and for you guys to come over here to give me the opportunity to talk about one of my preferred group of parasites, the Trichomonas. And I'll try to do that in the context of that is relevant for people interested in natural history. And I'd also like to thank Tom for the really nice talk he gave earlier on, that he sort of presented several places the importance of the environment in relation to human health, and more broadly speaking, health in general. And what I would like to do today, hopefully, is convince you that studying parasites, historically mostly focused on human ones, is much more interesting, much more valuable and relevant in multiple ways when we do that in the context of the relatives of those human parasites related to what's out there infecting other hosts. And in particular, when we look at pigeons. Now, I thought I'd give you a little background, so Gordon mentioned that a little bit. But since I have a bit of a strange accent, I thought I'd tell you why. Because I was born in Lausanne, so we speak French down there. And I was fortunate enough to study biology. I think I was a biologist since I walked, basically, looking at insects and so on. Mostly ants. I haven't seen many beetles, so I don't know much. But I do look at bees as well and try to wake them up when in the morning it's too cold and so on. So I was always a bit of a biologist, and I was very excited when I was able to go to university to study this. The initial plan was to do zoology and botany. And by some accident and the way the teaching was going on for those topics, I stumbled into molecular biology, cell biology, biochemistry. Okay, so I got diverted from my initial plans to do ecology. Then I did a PhD in molecular cell biology, but I was still a bit kind of connected with ecology, so I did some work with an NGO that looked at river, the lake, Lac Léman as we call it, to try to reduce pollution and so on. So I already had an interest in ecology. And then I moved into the Natural History Museum. There was another accident. I saw an advert just a couple of days before going to London for a postdoc at Natural History Museum. And I happened to have been to the museum quite a few years before that, and I always thought, what an amazing place. A cathedral for biology, that's the way I sort of described it. And for those who have been there, I'm sure that quite a few kids go there as well and so on. It's just a magnificent place in terms of architecture, but also in terms of all the records they have, libraries and so on, enormous collection. So I was fortunate enough to be able to work there and learn about microbial eukaryotes. I had this frustration of not doing much about evolution when I was in Lausanne, and I was bingo in the middle of an institution where I just looked at biodiversity, looked at evolution. It was heaven. And my initial work was on ciliates, so microbial eukaryotes. So we heard about the lack of interest, or sometimes the disgust about insects. Microbial eukaryotes are also kind of typically ignored and not very much looked into, unless they're human parasites, where people get a bit more interested and spend a lot of money in, but there's not much corresponding money put into understanding parasites of animals or microbial eukaryotes more generally and their role in the environment. So anyway, so on ciliates, I got interested in that, and that allowed me to combine evolutionary biology, molecular cell biology, and I got interested in parasites, in particular two groups, Trichomonas and Microsporidia, and today I'm going to talk about what I've done with Trichomonas. As my research was getting a bit on the French side of things, what we were doing at the Natural History Museum, I moved in Newcastle University in 2004, so just over 21 years over here. Initially, I was in the same school as Gordon, so we were colleagues teaching there. And basically what I worked on is the molecular cell evolution of Trichomonas vaginalis, a human parasite, sexually transmitted pathogen. Say a few words about that. And then we'll see how I got interested in relatives of those parasites. And that brings us to what we're going to talk about later on as well. Parasites infect birds, pigeons in particular. And then how they connect with other hosts. I also got interested in molecular basis of gut microbiota host interaction, really important component of any animal health. We live actually in the microbial world, okay? And we don't really function properly without those microorganisms. That's kind of an interesting or overlapping interest with the Trichomonas work, where these guys live in the same places. So these are organisms that live on mucosal surfaces. So these are sites where we have continuous, intensive microbe-host interaction, and they are central to many aspects of human health and disease. And since October, I took early retirement, and one of the ideas of the early retirement is to be able to change topic and go back to my environmental interest. So it was very nice to see Tom's talk doing a PhD on environmental issue, which was initially my initial dream. And then I got diverted, and hopefully I will be able to go back to that and do education and action on the field to hopefully help revive, improve the quality of the environment. And we heard from earlier presentation how we are very good at damaging it continuously, unfortunately. Okay, so we need to make efforts to reverse that to some extent. What I'll try to plan to do in this presentation is roughly three things: a bit of introduction about microbial parasites, a bit of semantics. Then I'm going to talk about Trichomonas, one of the best studied one, not say too much about it, but just mention a few things. And then we're going to jump into hopefully the things that get you more interested, where we talk about pigeons. And that includes, we're going to look at the diversity of pigeons. There's this magnificent book that I happen to have. I only bought the first 4 volumes. It was too expensive to buy the others. But all volumes are available at the museum in the Natural History Society library. So if anybody is interested in the pigeon I'm going to talk about today, or any other birds of the world, they are all described in here. It's just an amazing, amazing book. So I will show some images later on. Anyway, so let's keep going. Let's talk a little bit about microbial eukaryotes. They are of great interest to me, but they're interesting for many reasons. They're important as model system to study eukaryotes, molecular evolution. That was how I became sort of engrossed into studying them. Report in ecology, primary producers, and so on. Food industry, biotechnology, beer, bread, all of that depends on microbial eukaryotes. And of course, that's one of the things I spent quite a bit of time on, and I got funding from the Wellcome Trust to work on that, is the medically important parasites. But with the years, I became quite interested as well on those that infect animals as well. And hopefully at the end of that presentation, I will convince you that studying human parasites is much more interesting and powerful if we actually do that in sort of integrating the relatives among animals. Microbial eukaryotes come into tremendous variation, shape and sizes. Is a very nice figure for Blind Finlay, and he was a microbial ecologist, freshwater. All eukaryotes, what they have in common, they have these very complex endomembrane systems, like our cells, so we're all eukaryotes. So we are all related to each other, and we all have these very complex cells that allows these guys to function. The Trichomonas we're going to talk about are somewhere around these sizes here. Okay? But you get some big monster ciliates or amoebae that can be even sometimes seen with the naked eye. Okay, so Trichomonas. I got interested about them initially from an evolution perspective, a very interesting model to try to imagine how the early eukaryotic cells look like, how they function. They were thought not to have mitochondria. We showed that was not the case. They are symbionts of animals, humans. They are all extracellular parasites. Okay? So they live outside of our cells, tissues, but they live on the surfaces of mucosa, and they can interact intimately, as shown here. A nice scanning microscopy from a colleague in Brazil. These are vaginal epithelial cells, and this Trichomonas vaginalis in amoebae form actively interact like a pancake, if you want, and they can shift from a trophozoite, flagellated form, to these amoebae form. Okay? And in the process of doing so, they can cause damage, inflammation. Okay? So 2 species that are most studied are T. vaginalis, I just mentioned, but also the oral one, T. tenax. So I will briefly mention them, but then we're going to talk mostly about those that infect animals. Okay? And in particular, Trichomonas guyanae and relatives, and we'll see how diverse they are and how pigeons are important to understand those parasites. And understand not only parasites of pigeon, but our own parasites and parasites that infect pets, horses, and so on. Okay? So hopefully that will attract your curiosity and may stimulate some of you to look at this magnificent book the section on the pigeons in there. Okay, first I like to present them as symbionts. Some people use symbionts when they mean mutualist, so there's a bit of discrepancy from semantics. I like to use actually how Heinrich Anton de Barry, he was a German guy, but it's a French name. Apparently they all derived from a village in Belgium. So I bumped into one of his ancestors. That was interesting. From Switzerland, Basel, doesn't speak French name. I have a Germanic name that speaks French, so it was an interesting mix-up. And we met because I heard his daughter speaking Spanish with an Argentinian accent. My wife is Argentinian. Anyway, so interesting guy who is basically the modern founder of modern plant pathology and mycology, and he's the one who coined the term symbiosis. So different organisms living together, okay? And that's a general sort of context, and it's very nicely explained. Anyone who wants to read about this in this little paper here, that sort of goes back to the history of how it came about. So symbiosis, how different organisms interact, can have different outcomes. And you can have mutualistic interaction, both partners benefit, and then can become dependent on each other. You can have commensalism, one benefits, the other is not bothered at all, makes no difference whether they interact or not. And then there's what we call parasitism, one depends on the other one and causes damage when you take advantage of it. Okay? So I like to think about those interactions as symbiotic interaction, and you can have dynamic shift between these different outcomes. And the reason this might change is because life is complicated and it's going to be context dependent. Now, we like to talk about organisms, classify them, put into boxes. One of these boxes, there are books and there are lectures, there are courses organized, parasites. Okay, so we put them in the box parasites and that's it. But it's more complicated than that. And the outcome of host-parasite interaction depends on the context of this interaction, and that includes the characteristic of the environment. And we heard about earlier on the collapse of insects, pesticides and so on. Bees, for example, are much more susceptible to the neocorticoid we heard about earlier on if they're infected with parasites, with microsporia. So when companies test those chemicals, they use certain conditions. They say, oh, you see we can use those chemicals, the bees survive in our little boxes. It completely ignore the reality of the environment where they may bump into a pathogen like a microsporia, and if you expose those bees to those doses, they are dead. So it's completely irrelevant, those experiments they did in the lab. Okay? So that's the kind of things I think it's important to realize. You know, life is really complicated, depend on context, and I will give you some example of context here where, depending on what kind of organisms you have around, including what host you have and what microbiota, what microorganisms associated with them, will determine potentially quite different outcomes. So there's loads of parasites that you can have. This is a very nice textbook, Parasitology, a conceptual approach, and it has this nice overview. We're going to talk about two, Trichomonas vaginalis and Trichomonas tenax, and then that will be the basis for us to talk about then parasite infecting the birds, pigeons in particular. So these are extracellular parasites. They live specifically on the surface of mucosa, either from the STI, from the digestive tract, the oral cavity. They can infect sometimes the lungs, sometimes they infect also the salivary glands. They are associated with periodontitis and so on. Here's one image. This is a scanning microscopy of a parasite that we talk about, Trichomonas gainae. All these things called trichomonas, you can basically not make any you can't differentiate them when you look at the microscope. Okay? You need to do statistics, size, and so on. So we depend on molecular methods, and Tom mentioned that, an environmental DNA approach, where you can characterize what organism is present according to specific markers. And I will show you some example of phylogenies, phylogeny trees based on molecules that tells us, oh, this one is more closely related to this one and so on. Okay? And we're going to use that as the basis to understand how human infected parasites relate to animal and bird infected parasites in particular. Here's an example of a group of intracellular parasite. They need to go inside. I used to work on Microsporia. I mentioned that in the context of bees, for example. And the combination with pesticide is absolutely lethal to the insects. But let's forget about them and keep going with Trichomonas. And I like to think of the interest and importance of looking at this, and I think Tom gives a nice example of that, with a nice figure of how healthy environment, if you have a rich environment around you, and you have nice hedges, fields with flowers, forests and so on, that has also impact on human health. So people came up with this principle of the One Health concept. To me, that's just ecology. Okay? It's a fancy word to say ecology. It has an interesting PR aspect, I think, which is the health. We can all relate to health. And I think that is an important concept that we should hammer continuously to the government, to the developers and so on. If you're really thinking about or interested or pretend to be interested in human health, you've got to be interested in animal health, environmental health and so on. That has to be integrated to allow us to live in an environment which is not too polluted, where you can drink the water, where you can get food. Okay? And we had a nice, with Tom, the presentation about the importance of the pollinators. Okay? If we don't have those guys, we're not going to have the same quality of food, amount of food around us. So I think that the One Health concept is really interesting, and these are kind of the issues: growing human population, intense traveling, farming, large-scale food production, pathogens of farmed animal, pathogens of wildlife. We're going to say a little bit about that when we talk about pigeons. Pigeons are used in some countries quite a bit, not very much here, for food production. Say a few words about that later on. And then pathogens of wildlife, and then of course there are human pathogens of zoonotic origin. I'm going to talk about how Trichomonas can be traced back to animal, the one infecting humans to animals. So it's an example of zoonotic transfer. And then climate change, of course, is a big factor that impacts all of those three things. Us directly, the animals, the plants, etc. Okay. So we're going to look at a little bit. I got more interested in that because I think we need to think that way, both locally. I think there was a really important message from Tom. We need, all of us are involved with this, can do things. And at the end, I will give you a little bit of information relating to those pathogens that can be transmitted between pigeons and passerine, for example, on what we can do, how we feed birds in the gardens, for instance, and how we do deal with the issue with giving them water or food. These are potential sources of transmission of pathogens. We're going to see in a moment the relevance of that. So we are all involved, small scale, large scale, but hopefully I'll give you some interesting information related to that. So, most of my work initially was on Trichomonas vaginalis. Okay, so we're obsessed about our own health, obviously. And this is a parasite that is present in women, male, sexual transmission, and it goes through these different phases. It can be a trophozoite, an amoeba. It can eat things. Here's eating yeast. It can also eat bacteria. It can also eat our own immune cells, for example, illustrated here. So there's a lot of interest to try to understand how it can do that, how it binds to things, and so on. So I'm not going to say much about this. I would say a little bit about something really interesting about Trichomonas, is that they're not typically on their own. They live in a complex environment where we have human cells, but a lot of bacteria. But in addition to that, they also have endosymbionts. Two kinds. Mycoplasma. You may have heard of Mycoplasma. These are also potential pathogens. And some of them live inside the Trichomonas. And you can also have viruses, Trichomonas virus particles. That is potentially quite relevant and important for the pathobiology of this, because they modulate the inflammation that is triggered in our urinary tract, in particular in the women's system. And that is what can cause problems, not only pain and so on, but also issues with the embryo development, for example. So these are the kind of things we obsess about to try to understand what's the molecular basis of this. And we, for example, demonstrated that Mycoplasma, when they are present, change completely the set of genes that Trichomonas express in a quantitative way and qualitative way. I'm not going to go into detail, but the other thing we're interested in how they interact with the bacteria. And we know that they actually eat In this slide here, I want to mention Leukemia lactobacillus. It's a very important member of the vaginal microbiota that maintains health and makes that environment much more resistant toward a range of pathogens, including HPV, HIV, HSV, and so on. So if you have Trichomonas vaginalis, you typically have a loss of the lactobacillus population. Okay? So I think that's enough for me about this. The thing that we looked at, we have some couple of very nice PhDs around heart. We got this PhD this year, and we have a paper in press that describes the molecular details about how the Trichomonas interact with the bacteria and eat things. And what we've demonstrated as well is that these features in the human parasites are all shared with the parasite that infect birds. So what happens in the human was probably invented and put in place in the bird system. I'm going to say a few words about that. But I'm not going to bore about the details. The only point is that we found genes expressed by Trichomonas that correspond to enzymes that we know are derived from bacterial genes that then can target the bacteria. So it's kind of a warfare between protozoa and bacteria, which gene transfer events. So it's quite fascinating from an evolution perspective. The main take-home message here is that all the species we looked at have the same machinery. It's a conserved feature, and it's probably a conserved feature that was invented in pigeons. Okay? So, what I was really surprised when I was looking at the literature is that nobody really discussed the fact that parasites that infect humans in red, here's T. vaginalis on top, Trichomonas tenax, let's forget the others. These are isolates from human oral cavity or the urinary tract. Okay? And if you look at the phylogenetic relationship, which is a molecular phylogeny here, you basically compare sequence and see how things are more closely related, okay? Instead of using morphological character. And you can see that the various closely related sequences that were all isolated from birds. So everything in blue here comes from birds. And if you look at the literature, people completely ignore that. So I decided to look into this in more detail, and we wrote a paper with colleagues in the US that sort of summarized all of this information. And that got me really interested in studying the human parasites in combination with the one infecting the birds. Okay? And that's what we've been doing now the last few years. So, Trichomonas vaginalis, important human, that's actually the most common cellular STI on this planet. Not in the UK. In the UK, it's very much depending. Newcastle actually went to visit the STI clinic here, and they told me, no, we don't have that many of those. Okay? If you go to London and some Caribbean communities, the prevalence is quite high. Okay? If you go to some place in Africa, the prevalence is really high, and there's a direct correlation with how much HIV is around. So it's a really important contributor to HIV transmission. So it's relevant from that perspective, public health perspective. Trichomonas guyanae. So just to make sure we have that in mind, it's down here. Is there, can I use the, yes, I can use it. Can you see that? Yeah. So here we have, no, you can't see anything. Is that the laser?
We don't have a pointer, Robert.
I think that works, yes. So Trichomonas guyanae is here, okay? And here's Trichomonas tenax, the oral cavity, and then we have other Trichomonas that infects pigeons. We're going to talk about them in a moment, okay? So they're very closely related. So we worked on instead in Trichomonas guyanae, and there's quite a bit of work done on them as well, also from the genome perspective and so on. These infect pigeons, very common. They cause canker, so you may have heard of that. What it is, it's basically a nasty inflammation with accumulation of white blood cells and so on, and that blocks the digestive tract, so they can't drink and feed properly. Okay, so eventually they will be ill and die. In raptors, it's called France. Okay? There's also a case of epidemics where there was transfer from pigeons into finches. We'll talk about that a bit more. And then Trichomonas tenax, that's an interesting one. People thought, oh, it's a human one. Not really. If you look around, you find it in cats, you find dogs, horses, birds as well. We're going to talk about that in a minute. Okay? So interesting example of when we start to look into more detail how things are distributed across not only human population but also animal, we realize that actually we're quite ignorant. Okay? Okay, here's a case of, which bird is that? A little test for the Natural History Museum members. Peregrine, yes, a peregrine. One of their preferred food is pigeon. They will hunt pigeon like crazy. Okay, I actually seen that once in Cornwall. It was amazing. So there was a pigeon desperate. I don't know the outcome of the fight of the chase, but the peregrine was chasing the pigeon, and they were along the coast, and you could see them going up and then down the other side, and it happened 3 times. Absolutely amazing to watch. In this case, the pigeon were caught will be eaten. So raptors, such as peregrine that eat pigeons, can get the infection that the pigeons carries. Okay? And that's called France. Here's a bit of the data for Trichomonas tenax, where you can find it. So initially, as I told you, found in human, but we know it's now in dogs, cats, horses, and also wild birds. So there's quite a bit of data on that. Here's an example of data from pigeons from Austria. I actually met a colleague, Ivana Bilic, who contributed to that work. And here's a study by French and Belgian people who showed that if you sample, chromatal sample from these different birds going around water surfaces, you can find very similar sequence, if not identical, to Trichomonas tenax, suggesting a broad distribution of Trichomonas tenax and Trichomonas tenax-like, okay, including in pigeons. We're going to go back later on to that. So basically, that got me interested in other as well. So here you have a colleague from Czech Republic, Jan Takezi, with whom we collaborate on different species now, including species isolated from horses, dogs, and cats. And so there's a really broad distribution of these guys, and they're all associated in the context of the oral one with periodontitis. Okay. Here's an example of a new species that were completely, we didn't know it before sequencing took place. This is I keep forgetting its name. Band-tailed pigeon. So it's the biggest pigeon in North America. It's very common along the Californian coast, and they also migrate along down South America. And they are under massive pressure because of change of landscape and so on, but also habitat issues. But there was also a collapse of population due to an infection that was really nasty due to Trichomonas stegleri. So it's a new lineage, clearly distinct from T. gallinae, which is over here. Okay? And it's really closely related to Trichomonas tenax. So you can see that this infects birds, leads to similar disease than Trichomonas gallinae in the pigeon, so canker. I'll show you a picture in a moment. And they're really closely similar to what infects humans. So here's an outcome of one of the infections. These are the characteristic features of canker. So massive accumulation of immune cells, inflammation, and that completely blocks the digestive system of the bird. What about the UK? So in UK there are 5 different species of pigeons, okay? All illustrated here. I see this one on a regular basis in our garden, okay? It's actually the most common. I didn't know that. I thought it was this one. But in terms of numbers, apparently that's the most common in the UK. And if you ask many people, they say, oh, they're just a bit boring birds and flying rats and all of those expressions that people have, right? So we're going to look in a moment that if you look at pigeons across the world, it's actually much more fun than that. And as I said, maybe at the end of the presentation, hopefully it will convince you that you should have a look at this book. Now, one pigeon species is interesting. It's actually in French we call tourterelle turque, the Turkey dove, because it used to live down there until 1900. And then suddenly migrated like crazy. And you find it all over Europe now, including the UK. And you can find it in North America as well. Okay, so it's a big guy that traveled all over the world and having fun doing so. All Trichomonas gallinae. Okay? So all the red arrows highlight sequences derived from isolate coming from pigeons. I highlighted in black the one that comes from human, tenax, or mammals, sorry, and dogs for here, and here we have Trichomonas vaginalis again. Okay? All the others are from birds. This is non-pigeon birds, so here we have a raptor, and here we have passerine. We're going to talk about passerine a bit later in a moment. So all of those species the 5 species that we find in the UK are all known to carry different lineages of Trichomonas guyanei or relatives to it. Okay, so it's very common, and you can see that in these sets of sample, they were very common. Most of the species carry that stuff. So in that sense, it's probably not just a parasite. It might be part of the system, and that most of the time it's not causing disease, but when the condition changes or when the bird is stressed, it might cause disease. Now, a dramatic case of transfer of host, and clearly leading to a mismatch between the host and the parasite, is transfer from pigeons to finches, greenfinch, chaffinch. Here's some example. It even made the BBC. Disease threatened UK gardens, deadly bird disease Trichomonas is spread to Europe. So not only the transfer occurred in the UK, it spread very quickly across Europe, Scandinavia and so on. Okay? So really impactful events of transfer of hosts. And you can find some interesting information in the RSPB. Keep your garden birds healthy and provide advice. Okay? And I have a PDF from another organization. If you want, anybody wants to email me, or if you check, you can find this as well yourself. So what to do to reduce the chance for these transfer to happen. Because most of those transfers are thought to take place because of the interface between our environment, humans, and bird environment. A lot of birds have, of course, adapted to the presence of gardens, where we put food and water to provide drinking source of water. And this is probably where those transfer events took place. Okay? Because a pigeon and a chaffinch, they don't really talk to each other, they don't meet very often in the wild, they occupy completely different niches, they feed on different things. These were quite dramatic events. So in 2005, there was a clear decline of the population of the greenfinch, similar event, a big delay for the chaffinch, and these are all associated. So all these dead birds that were found, the collapse are associated with, all of that was associated with lots of dead birds, and they were all infected with Trichomonas guyanei. Okay? So these are transfers that took place between pigeons, and here we have actually a chaffinch and a greenfinch fighting for water access. And this is one of the potential sources. And people have done experiments demonstrating that if you put pigeon with infected Trichomonas accessing the water, the Trichomonas can survive long enough, because typically it's full of organic matter. As long as you have that bacteria, and Trichomonas can survive a bit, and that can lead to transfer to new hosts, such as these birds. What about farming? I mentioned farming earlier on. The pigeons can be eaten, and some people love eating that stuff. I never eat any pigeon. I was told it's very tasty. And if you read a bit of literature, it's actually quite healthy meat as well. It's very lean, and the Chinese in particular use that as part of the diet for Chinese medicine. So if you have certain conditions, they will advise you to eat pigeon to help you. A bit like having chicken soup for flu. Okay, same principle. All right. They make a lot of meat from pigeons. So here we have some information here. They had 100 million pairs. They had 1.6 billion squabs that were used to make meat. So it's a big thing. And they were interested in studying the relationship between Trichomonas guyanei infection and how the microbiota changed. Remember I told you about Trichomonas vaginalis and loss of lactobacillus. There's lots of other things going on, but that's one of the key things. So they were interested to they asked the question, and we wanted to do that initially, but they beat us to that. We had lockdown, we couldn't do any work at the time, and they published this paper that was really exciting for us because we were interested in that, and we eventually collaborated with them. Give a few more things, information later on. But to make a long story short, basically what you observe in human with the collapse of lactobacillus population and change of the composition of the microbiota present in the human system, you say something quite similar in the bird. So when Trichomonas guyanei infect pigeons, they will lose lactobacillus, for example. Okay? So we kind of analyzed that. And then the other thing that was really intriguing that we discovered by studying a Geordie pigeon. So Nick Bailey, PhD student in my lab, very talented guy. We were planning to do the experiment the Chinese did, but then we were having some interesting data that came out with the data set that he had put together. So we spoke to to a pigeon fancier, so it's quite a few people doing pigeon fancier around here. And so we had access to that, and we helped him to establish which pigeon were positive. There were quite a few for Trichomonas guyanae, and they tend to treat them if they are positive, okay, to avoid the calculate. And what was really interesting, we found out that in one of the pigeons, I'm not going to explain how we found it because it's complicated, but we identified a Mycoplasma species that we call Trichamica, that loves Trichomonas. A bit of Latin here. Nick came up with a name. I challenged him, find a good name for your new species of Mycoplasma you found. So he came up with that name. And it's very closely related to something called Mycoplasma guerreri. Now, why is that interesting? This you find in the human system. And you find it only when you have Trichomonas vaginalis, because they are obligate synmates. So we've discovered a bacteria very closely related to a human Mycoplasma that is associated with Trichomonas, and Nick could demonstrate that the Mycoplasma, this one here, is strongly associated with the Trichomonas guyanae. We basically found it almost exclusively, not completely, when Trichomonas guyanae is present. So it's a very interesting, surprising result, suggesting that the symbiosis that we thought is important in the human system and that modulate the pathobiology of Trichomonas is actually, there's something similar going on in the pigeon as well. This is to show that Lactobacillus also population collapse in the presence of Trichomonas guyanae. And then this is data from the Chinese. So we could find the Geordie pigeon had that Mycoplasma, and the Chinese pigeon has that as well. And we worked with a colleague in Sardinia, and they found as well this in pigeon in Sardinia. So it's basically global. So Trichomonas guyanae is everywhere, and that new species of Mycoplasma is also everywhere. So that looks like they have a coevolution event between Trichomonas and Mycoplasma, and then when there was a transfer between birds to human, that transfer involved two things, a Trichomonas and a Mycoplasma. Potentially, that was actually facilitating transfer, because what we demonstrated is that when Mycoplasma is present, the transcriptome, the set of genes that the parasite express, are very different and make it a bit more capable, for example, of binding to human cells. So maybe something similar happens here. So quite intriguing data, and that illustrates the interest of studying pigeon. Now I'm going to divert a little bit, and I'm going to present just sort of a general, very briefly. I don't pretend to know any of this about that, but I love the picture of the book, and it was an interesting thing for me to discover, because I always thought, pigeon would be boring. There's so many birds that are much more interesting than pigeon. But in fact, when you start to look into this, I was completely mistaken. And hopefully at the end of the few slides I will show you now, I will convince you that pigeon is actually quite exciting and interesting, not only to study parasite, but also because they have a tremendous diversity of those species, and some of them are beautiful. So if anybody is interested, the Great North Museum Library have all the books of that handbook of the birds of the world. I was only able to afford the first 4 volumes when I was young. Later on, I was not allowed to buy any more copies. But if I'm desperate, I can go to the Great North Museum Library. They're all there. Okay. Okay. So let's a few pictures from that book. First thing, it's a really nicely organized book. So it gives a general introduction about the biology of these. Note there are more than 300 species of pigeons. I had no idea about this. And then, if you look at the diversity of them, 2 major groups, the Columbinae and the Trerorinae, so 123 species here, almost 200 species there, and the rest is just a few species. Okay? These are so-called fruit dove, so they specialize in fruit, and that is it's a bit of everything, some of them fruit and so on. So, nice diversity, and some of them look really nice. So, the New Zealand pigeon, Enifaga Noveozelandia. I like to say Kerulu. Kerulu is the Maori name. So I went recently to New Zealand. I have a colleague with whom I collaborate for many years on Trichomonas. And he organized an international conference that I organized, the equivalent here in Newcastle, by the way. But anyway, so I was very excited to go to New Zealand. I always wanted to go there. And one of the great pigeon here is called the keruru, and I'm going to see in a moment, this also carries Trichomonas, but a very distinct one. Then there's some other things. This is from the Comoro Island and Seychelles, beautiful pigeons, so far from being boring, and definitely not look like flying rats. Some more images, just a few examples. I just took a few cases. So you can see there's tremendous diversity in terms of colors. Some of them are really elegant with the orange stuff here. Nice combination of colors that obviously we can't see here, but you know, if you travel a little bit, you can see all these wonderful different species of pigeon. So why is that interesting in the context of this presentation? An Australian group decided to ask the question. I'm okay with the time? Sorry, go on.
About 10 minutes.
Okay, it's fine then. And they decided, okay, so we've got this tremendous diversity of pigeons. We know that Trichomonas gallinari and some other lineages are present across several species that people looked at. What about the distribution of Trichomonas? Do we find them in this different set of species that actually correspond to these major lineages I just told you about earlier? Okay? And so a really interesting paper, so we were really excited when I saw that paper. So they sampled all these different pigeons. You can see this one here corresponds to a distinct lineage here, the so-called crowned pigeons. So they have species from across here and species from across here, including some of that. So really across the global diversity of pigeons from across the planet. So that's the sampling. And it's all pigeons from New Zealand, New Papua New Guinea, including the keruru from New Zealand. Okay, so that's a bit of an outlier here. And what they discovered was really exciting from a perspective of biodiversity, parasitology. That was great. All these lineages that are not highlighted by colored arrows are from these different pigeons. So the diversity dramatically was boosted. So once they decide to look at diversity of these pigeons, can we find sequence related to Trichomonas? The answer is yes. Not only that, but you basically more than double the diversity of what we thought we knew about Trichomonas. So really interesting correlation between pigeon diversity from across different countries, islands, and so on, and the diversity of Trichomonas lineages that you can find among them. In red, I highlighted the one that we found in mammals. Trichomonas vaginalis, human. Trichomonas brixi, there was a lineage discovered by Honsa's lab in cats and dogs, oral cavity. Trichomonas tenax, that's human, cat, dogs, and birds. The keruru, really nice pigeon here by the way, has a very long branch, probably because it's separated from all the other species a long time ago when New Zealand was formed. And that species is specific to New Zealand. So there's a correlation between distant relationship, speciation event from the host that relates to differentiation of the parasite that co-evolved with it. Okay? Then a vulture here, this was discovered in one of the vultures that you find in Spain. I forgot the name now. Maybe whoever is an ornithologist might remember. I think it's the Egyptian vulture, isn't it? Does anyone remember vulture species? I think it is. It's one of the guys, a bit whitish. It's not a big size. Anyway, so that was discovered in this one, and that's probably a vulture feeding on the dead pigeon. Okay? So another example of transfer. So really interesting case where when you start to ask questions about when you consider biodiversity, in this case of pigeons, how you can have a better understanding of the evolution and diversity of the parasite, and that really strongly further highlight the hypothesis that the parasite we find in human, cats, dogs, and so on are all derived from pigeon-infecting species. So we have multiple host transfer that took place from pigeons to human, pigeon to cats, pigeon to dogs. Okay. So, be careful with who you kiss, right? And when you drink the water, okay? So, we've discussed the example of transfer between pigeon and finches. This boy clearly likes pigeon. Might not be a good idea to do that. Here I have a picture, I put that on purpose. My wife loves dogs, and I'm not really keen to have dogs, it's too messy for me and everything. But anyway, so I refuse to have dogs at home, but I'll do dog sitting. Okay? And she always does that. And she does that on purpose, smiling at me. And it drives me mad. He said, look, don't do it. But anyway, it's very funny, it's an internal joke. So, but just don't do it. You may get Trichomonas tenax that way. And if you have peritonitis, you definitely don't want that, because Trichomonas tenax is associated with a worsening in the diagnosis. Okay? And it's probably due to the fact that it reinforces the inflammatory process by feeding on bacteria, making the microbiota more dysbiotic, etc. I don't go into detail, but basically the inflammation, which is the key feature of peritonitis, got worse. And indeed, there's a very strong correlation between worsening of prognosis, and Trichomonas tenax. I'll also show you transfer of falcon, for example. That's probably how these guys get them as well, got them initially, and eventually became within cats, within dogs. There's also evidence in horses, and there's some other species I haven't mentioned that are also present in horses. We had some data on these as well. And all of that eventually led to Trichomonas tenax in human and T. vaginalis. And T. vaginalis, as I said, is particularly relevant in context when you have high prevalence of HIV, and those two things are clearly impacting each other. So again, we need to be careful how we interact in our environment. We need to be careful what we do in our gardens. So if you have little buckets of water that you intend the birds to use for drinking, I will clean that every single day. Otherwise, you're going to allow pigeons to infect them, and pigeons have quite a high rate of Trichomonas gallinae. And if that jumps in the wrong, it's actually only some strain. All these strains that kill those guys is one lineage. They're called A1, which is really nasty. The others are not. So there's variation of that. But if you happen to have a pigeon with an AY strain and it jumps into one of those birds, it can have dramatic impact. And there's advice provided by RSPB on how to deal with that. One of them is to clean the water things. And also do not use flat surface area where the food sits, because that allows the detritus to be accumulating and help transfer of infection. And I think with that, we are done. Oops, what the hell are they? So I just want to show the last slide. Here we go. Just to acknowledge the people that I had the privilege and pleasure to work with. Is it working? No, it's not working. I have no idea what I pressed. University computers. Brilliant, thank you very much. So, I mentioned Nick, Lenshina as well, Adam. These are PhD students I really had a great pleasure to supervise. We all worked on Trichomonas projects together. Lenshina did quite a bit of work on Trichomonas tenax, Nick on Trichomonas gallinae. Adam took over and after Nick and worked on the Trichomonas gallinae, where we demonstrated and he found new sets of genes and new activities that explains how Trichomonas actually deconstruct and eat bacteria, and that's directly relevant for inflammation. More recently, Isra Asker, she's now finishing her PhD. We'll write up by the end of the year on the oral ones from dogs, human and cats. And we also did a lot of work with Valentina and Gigi, so they're based in University of Sassari. They had a lot of interest in studying Trichomonas mycoplasma and showed that the combination of Trichomonas mycoplasma changed completely the behavior and the pathobiology. And I think I'm going to stop here. Thank you very much for your attention.